Alzheimer's disease is a neurological disorder that progressively interrupts memory and thinking skills, and eventually the ability to carry out simple, everyday tasks. There are two main types of Alzheimer's, and their main difference is at what age they present themselves. Early onset Alzheimer's can occur as early as 30 years old to 60 years old, while late onset typically occurs after 60 years of age. The majority of people with Alzheimer's disease have late onset Alzheimer's disease. The discovery of Alzheimer's occurred in 1906 when Alois Alzheimer, a German neurophysiologist, discovered peculiar plaques in the brain tissue of one of his patients that had exhibited memory loss, paranoia, and confusion. Things we know now as typical symptoms of Alzheimer's. He found these same plaques in individuals with the same family of symptoms, and upon more research, was able to characterize and formalize the diagnosis of Alzheimer's. If we take a deep dive into the micropathology of Alzheimer's, there are two main signs of it in the brain, which are neurofibrillary tangles, or NFTs, and beta amyloid plaque formation. Neurofibrillary tangles occur inside of the neuron body and are a result of a protein called tau misfolding and obstructing proper cell function and communication. Healthy neurons, in part, are supported internally by microtubules, which help guide nutrients and molecules from the cell body to the axon and dendrites. In healthy neurons, tau normally binds to and stabilizes microtubules. However, in Alzheimer's disease, abnormal protein synthesis causes tau to detach from microtubules and stick to other tau molecules, forming threads that eventually join to form tangles inside of the neurons. These tangles block the neuron's transport system, which harms the synaptic communication between two neurons. As we can see here from this diagram, the hyperphosphorylation of the tau proteins causes them to detach from the microtubules and then, in turn, accumulate into neurofibrillary tangles. We can also see two images of the neurofibrillary tangles under the microscope. Next, we have beta amyloid protein plaques. So these protein plaques involved in Alzheimer's disease come in different molecular forms that collect in between neurons, as opposed to the neurofibrillary tangles which occurred inside of the neurons. So it is formed from the breakdown of a larger protein called the amyloid precursor protein, which we can see here in the second column of this diagram. We can see here from the amyloidogenic pathway that the beta secretase enzyme cleaves the amyloid precursor protein into two parts, which includes the C99. And then the gamma secretase further cleaves this into the amyloid beta protein and AICD. The amyloid beta protein then further clumps through amyloid beta oligomerization and then forms the amyloid beta plaques, which are what is found in this image to the left, which shows the interneuron space in the brain, where these plaques are forming and preventing proper cell communication. However, what does this have to do with neurotransmitters? As we know, neurotransmitters are chemicals that carry messages between two neurons to trigger an excitatory or inhibitory response that elicits a certain function. This, we know, is called a synapse. If the brain produces too much or too little of a particular neurotransmitter, problems such as memory impairment, confusion, and even depression can occur. In Alzheimer's, the beta amyloid plaques found outside of the cell body have been shown to increase the activity of acetylcholinesterase, the enzyme responsible for hydrolyzing acetylcholine. The increased activity of this enzyme, in turn, decreases the availability of acetylcholine, which is known to be necessary for memory storage, and other excitatory functions around the body, like muscle movement and learning. A decrease in this neurotransmitter can, of course, create a decrease in the brain's ability to perform these functions. As we can see from this diagram, the acetylcholinesterase, which is the red squiggly thing, is cleaving the acetylcholine before it is even able to reach the postsynaptic terminal. This decrease in acetylcholine is, of course, going to decrease the effects that the sending neuron intended to transmit, thus causing these lapses in memory and in learning. Now, the other neurotransmitter involved in Alzheimer's progression is glutamate, which is another excitatory neurotransmitter that plays a role in arousal, learning, and memory encoding. As neurons die as a result of interruption by plaques or neurofibrillary tangles, their main response is to produce and release glutamate as a sort of lifeline to the cells around. However, when there is mass cell death, which occurs in Alzheimer's and, of course, other neurological diseases, all of these cells are releasing glutamate, and it can actually produce an effect called neuronal excitotoxicity, which results from having too many ions floating into the neuron due to being overstimulated. This causes the same cell to die and causes a positive feedback loop where more and more glutamate is released and more cells die. As we can see from this diagram, this excess glutamate is making its way towards the NMDA receptor and increasing the calcium concentration inside of the neuron body. This increased or excessive increase of calcium ions actually disrupts mitochondrial function and decreases the amount of ATP available in the cell. This decrease in ATP in turn produces necrosis or apoptosis, which is programmed cell death. This in turn would release more glutamate, which continues the positive feedback loop. Currently, the ways to treat Alzheimer's revolve around mediating these two neurochemical pathways specifically. Acetylcholinesterase inhibitors, which we can find on the left, prevent the increased breakdown of acetylcholine, which we can see from the diagram that acetylcholine is able to make its way into the postsynaptic neuron in higher concentrations. To the right, we can see NMDA receptor inhibition, where glutamate actually still binds to the receptor, however, calcium is not allowed in, thus preventing the apoptosis and necrosis that we saw in the previous slide. Understanding neurochemistry is key to our understanding and development of cures and treatments for neurological disorders, as it provides the most straightforward pathways to mediating symptoms of neurodegeneration by providing targeted specific treatments. I hope you enjoyed my video, and let me know if you have any questions.